Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, materials science, chemistry, and physics, all told as compelling, easy-to-follow stories.
Glad to be here for this one.
Yeah, and today we are starting with a situation that I think you can probably relate to. So imagine you just saved up and you bought a brand new, incredibly complex piece of technology.
Oh, like a high-end smart car or something.
Right, exactly. One of those fully computerized smart cars. So it arrives, you're thrilled, but there's a massive problem.
Let me guess. It's broken.
No, the car is fine, but the only instruction manual it comes with is for a completely different vehicle, and it was made on another continent.
Oh, wow. Yeah, that is a problem.
Right. If you try to fix your car
Right.
or update its software using that manual, you're going to break something.
You're definitely going to break something. I mean, it is a guaranteed disaster. You might be able to, change the wiper blades because a wiper is just a wiper.
Yep.
But the minute you touch the intricate wiring harness,
Right.
or you try to recalibrate the battery system with the wrong manual, you are just working blind.
Which brings us to the strange question at the heart of today's deep dive. And here's a strange question. Why does a standard dose of a perfectly common anti-seizure medication work like a charm for a patient in New York, but cause a severe, life-threatening skin reaction for a patient in Hanoi?
Yeah, it's terrifying. And it is a real reality for clinical oncologists and neurologists globally. For a long time, these varying reactions to standard medications across different populations, they just remained a genuine medical mystery.
So today, our mission is to explore the hidden blind spots in modern medicine. We are going to look at how a massive new scientific effort just uncovered millions of biological secrets that could fundamentally change how we treat disease globally.
It really is paradigm shifting.
Yeah, we are exploring the myth of standardized medicine and uncovering what actually happens when the global medical manual turns out to be missing millions of pages.
Right. And to really understand how big this blind spot is, we have to look at the foundational problem geneticists have been grappling with for the last 2 decades.
Okay, set the scene for us.
So when the human genome was first mapped back in 2003, the scientific community hailed it as this ultimate triumph. We had essentially created a master reference manual for human DNA.
The manual for the human car.
Exactly. And the entire global medical apparatus, I mean researchers, doctors, pharmaceutical companies, they all started using this one database to understand diseases and design drugs.
But there was an underlying assumption baked into that celebration, wasn't there? The idea that since humans share about 99.9% of our DNA, a generalized reference genome would just be good enough to treat everyone on the planet.
Right. That was the prevailing thought.
Okay, let's unpack this because I want to push back on that for a second. If we are actually 99.9% identical, how much can missing one demographic really matter for day-to-day medicine? I mean, a heart pumps blood, a liver filters toxins. Why does that 0.1% matter so much?
That is the exact logical trap that much of the medical community fell into. The issue is really about scale and function. You have to remember the human genome consists of over 3 billion base pairs.
3 billion, wow.
Yeah, so that 0.1% difference, it still translates to millions of distinct genetic variations.
Oh, right, math.
Exactly. And more importantly, that tiny fraction contains the highly specialized instructions for how a localized population adapts to its specific environment.
Wait, so what kind of instructions?
Well, it dictates things like how your specific immune system identifies foreign pathogens, or how your enzymes metabolize complex synthetic drugs, even how your cellular pathways respond to local diets.
Oh, I see. So the 99.9% builds the car, but the 0.1% writes the software that actually runs it in a specific climate?
That is a great way to frame it. And the massive flaw in our global medical manual is that the genomic databases used to write the software are just overwhelmingly skewed.
Skewed how?
Historically, around 80% of the participants in genome-wide association studies, which are the data sets that tell us which genes cause which diseases, they are of European ancestry.
80%.
Yeah. Meanwhile, Asia, which holds roughly 60% of the entire world's population, has been severely underrepresented.
Okay, so let's use Vietnam as the focal point here because it highlights this discrepancy perfectly. Vietnam is the 16th largest country in the world. It has a population of about 100 million people.
Right, it's huge.
And because of its location in Southeast Asia and a very complex multi-millennial migratory history, it contains an incredibly deep well of genetic diversity. Yet, before the research we're digging into today, the standard global database, which is the famous 1,000 Genomes Project, it only contained 99 Vietnamese genomes.
99 out of 100 million people.
Yeah, wait, I'm confused by this. If the 1,000 Genomes Project was meant to be the global standard, why would they only take 99 samples from a country that big? Was it like a funding issue or just a huge oversight?
It was a combination of things, mostly historical infrastructure biases and a really flawed sampling philosophy. Early genomic projects relied heavily on existing research hubs, which were, primarily in North America and Western Europe.
Sure, where the big labs are.
Right. And when they did sample other regions, they often took this representative approach. Those 99 Vietnamese individuals, they were all sampled from one specific urban area, Ho Chi Minh City.
So they just assumed that one localized cluster could serve as a proxy for the entire nation.
Exactly.
But statistically, I mean, polling organizations can predict a national election with 1,000 people. Why couldn't they capture Vietnamese genetics with 99?
Because human genetics isn't a binary vote. It is this unfathomably complex network of rare variants. They are shaped by thousands of years of regional isolation, topography, and migration.
So you can't just poll one city block.
No, you cannot capture the genetic diversity of isolated highland populations and coastal communities by only sampling people in one major southern metropolis.
Right.
By relying on that tiny localized sample, scientists realized they were just flying completely blind in Southeast Asia.
So how did scientists figure this out? This is where the story gets really interesting. We are diving into a landmark study published on July 22, 2026 in the journal Nature Communications.
Such a huge paper.
It really is. A massive team of researchers, led by Dr. Nam S. Vo and Dr. Quan Nguyen from the VN1K Big Data Research Institute, alongside other global institutions, they basically decided to completely rewrite the genetic map for Vietnam.
Yeah, they launched what's called the VN1K Project, and they designed it specifically to overcome the flaws of those past studies we just talked about.
So what did they do differently?
They sequenced the genomes of 1,011 unrelated healthy individuals. But crucial to your earlier point about polling, they didn't just stay in a city. They gathered participants from 53 of Vietnam's 63 provinces.
Oh, wow. So they actually captured the true topographical and ethnic diversity of the nation.
They really did.
And the methodology they used is fascinating because they didn't just take a surface-level look. The paper emphasizes that they used high-depth whole genome sequencing, specifically 30 to 60x coverage, and they combined short-read and long-read sequencing technologies.
That is the technical magic right there.
Right, but I have to admit, those terms sound a bit opaque to me. What is the actual mechanical difference between short-read and long-read, and why did they need both to do this?
Okay, think of reading the genome like trying to reconstruct a massive encyclopedia that has been run through a paper shredder.
Okay, visual tracking, shredded encyclopedia.
Right, so short-read sequencing is like taking tiny shredded fragments, maybe a sentence at a time, and reading them with near-perfect accuracy. It is phenomenal for catching a single typo in a word.
Okay, but what's the downside?
Well, if an entire chapter has been duplicated or moved to a different volume, you wouldn't necessarily notice, because you are only looking at isolated sentences.
Oh, I see. So you get the micro details, but you lose the macro structure.
Precisely. Long-read sequencing, on the other hand, allows you to read massive, contiguous chunks of DNA. We are talking entire paragraphs or pages at once.
So why not just use that?
Because the error rate for individual letters might be slightly higher. But it gives you the overarching architecture of the genome. By combining both, the researchers achieved something unprecedented. They got the perfect spelling from the short reads and the perfect structural architecture from the long reads.
And that structural architecture is critical, right? Because it exposes the massive limitation of the old reference map, which geneticists call GRCH38. That global standard map is linear.
Yes, linear, like a straight line.
But a linear map forces everyone's complex genetic variations onto one single straight track.
And that is the fatal structural flaw of a linear reference. If a specific population has evolved an entire sequence of DNA that simply does not exist in the European reference genome, like an insertion or a complex structural variant, a linear map cannot accommodate it.
So what does the linear map do with the new data?
It just drops the data. It treats it as an error or an unmapped read.
That's wild. It just ignores it.
Yeah. So to solve this, the VN1K team threw out the linear concept entirely. They built a pangenome graph reference, known as VPR, utilizing a deep learning AI framework to analyze all this new data.
So mechanically, how does an AI-powered pangenome graph handle data differently than the old linear map?
So instead of a single straight line, a graph reference uses nodes and edges. It operates as a complex network.
Okay.
When the AI analyzes a genome and finds a structural variation, like a sequence completely unique to the Vietnamese population, it doesn't discard it. It creates a branching path, a detour, that splits off from the main reference and then reconnects later.
Oh, so instead of a flat paper map of a highway where you miss all the local detours, this AI-powered pangenome graph is like a 3D Google map showing all the alternate routes, overpasses, and hidden alleys in the Vietnamese genome.
That is exactly it. It maps everything without forcing them all to overlap on the same road.
That's incredible. And the results of using this graph-based approach were staggering. I mean, when they ran this high-depth data through their AI framework, the researchers uncovered 42 million genetic variants within this population.
And here's the real wow moment. When they cross-reference those 42 million variants against massive global databases like GnomeAD and the 1000 Genomes Project, they found nearly 8.5 million variants that were completely novel.
8.5 million. Wait, really?
Yes, millions of pieces of human biological data just completely invisible to global medicine until this study.
Entire localized evolutionary pathways that simply had never been recorded before.
Never.
Which brings us back to the mystery from the top of the show, the seizure medication. How do these novel variants explain why a drug acts like a cure in New York, but a poison in Hanoi?
So the researchers focus heavily on pharmacogens. These are the genes responsible for synthesizing the enzymes that dictate drug metabolism. They analyzed a specific genetic allele, which is just a variant of a gene, called HLA-B15.02.
And this allele is the culprit behind that horrific reaction to standard anti-seizure drugs, right? Like carbamazepine.
Yes. The condition is called Stevens-Johnson syndrome, and the mechanics of it are deeply alarming.
Yeah.
The HLA gene family is basically responsible for creating proteins that present antigens to the immune system.
Like little security guards.
Yeah, they are the molecular guards that hold up a flag saying, hey, this cell's infected, destroy it. But when a patient with the HLA-B15.02 variant takes carbamazepine, the drug physically binds to that specific protein and alters its shape.
So the drug changes the flag the guard is holding.
Exactly. The immune system suddenly misidentifies the altered drug-protein complex as this massive system-wide foreign invader. It launches a severe cytotoxic attack against the body's own tissues, causing the skin and mucous membranes to literally blister, die, and peel off.
Oh man, that is horrifying. It's an autoimmune cascade triggered by a common prescription. Now, in European populations, which is the people the global reference manual was built on, this allele is practically non-existent. A doctor prescribes the drug works, everyone goes home. But what did the VN1K Project reveal about this allele in Vietnam?
They found that this specific high-risk allele is carried by over 23% of the Vietnamese individuals in this study.
Nearly a quarter of the population, they're just walking around with a genetic trigger for a fatal drug reaction, and the standard medical manuals weren't even flagging it.
It gets worse. The study also revealed that over half of the Vietnamese population are classified as intermediate metabolizers for certain pharmaceutical compounds.
What does that mean in practice?
It means their liver enzymes break down drugs at a significantly different rate than the European baseline. So if you use the standard Western dosage guidelines, you're either going to underdose the patient, making the drug ineffective, or overdose them, causing severe toxicity.
So what does this all mean for the listener and for the future of medicine? I mean, the implications of this research are incredibly immediate. This isn't just theoretical data mapping. This directly changes clinical care tomorrow.
It absolutely transforms clinical screening. Let's look at prenatal screening. Expectant parents routinely take genetic tests to see if they carry pathogenic variants that could affect their child.
Right, very common.
But those standard tests are built on the old Eurocentric databases. The researchers analyzed a gene called PMFBP1, which is strongly associated with certain genetic disorders.
And what did they find?
They found that pathogenic variants of this gene are highly prevalent in Vietnam, occurring in over 14% of the population. But in European populations, the occurrence is less than 0.5%.
Which means standard Western prenatal screens aren't even looking for the diseases that actually threaten this specific demographic. They're running a diagnostic test for the wrong car.
Exactly. The VN1K database gives regional doctors the ability to finally calibrate their tests to the actual biological reality of their patients.
But the researchers realized that simply having the structural blueprint of the DNA wasn't enough, right? The environment plays a massive role in how these genes express themselves.
Yes, the epigenetic twist. This introduces the field of epigenetics, specifically a mechanism known as DNA methylation.
Okay, nature versus nurture. But if genes are just a static blueprint, how do things like the high rates of liver disease in Vietnam actually interact with or change the DNA?
Think of the DNA as a piano keyboard. The keys, the genes, are static, they don't change. But methylation is like putting heavy tape over certain keys so they simply cannot be pressed.
Oh, that's a great visual.
Biologically, the body attaches chemical tags called methyl groups directly to the cytosine bases of the DNA. This physically blocks the cellular machinery from reading that gene, effectively turning it off in response to environmental factors, diet, or stress.
So they didn't just map the keys of the genome, they mapped where the tape was placed.
And they found a fascinating epigenetic signature. A specific gene called DLGAP2 was found to be highly hypermethylated, meaning heavily taped over and turned off in Vietnamese populations when compared to Europeans.
And what does that gene do?
It is intricately linked to lipid metabolism
Yeah.
and hepatic function.
Hepatic meaning liver. So the high incidence of liver-related metabolic issues in the region isn't just about having different genes, it's about the local environment actively suppressing the genes that regulate liver function.
Right. The static genome only tells you what the body is capable of. The epigenome tells you what the body is actually doing in its current environment.
Speaking of the environment interacting with our biology, we have to talk about the hidden virus. Because while the team was sequencing this massive data set, digging through the deepest parts of the pangenome graph, they found sequences that were not human at all.
This is one of the most remarkable discoveries in the study. While analyzing the raw data, they detected genetic sequences belonging to the hepatitis B virus integrated directly into the DNA of nearly 10%, specifically 9.8%. Of the ostensibly healthy samples.
Wait, really? Not just floating in the blood, but literally integrated into their genome. How does a virus even do that?
Viruses like hepatitis B are insidious. To replicate, they can actually physically break the host's DNA strands and splice their own viral genetic material directly into the human chromosome.
Wow.
Yeah, and then the host cell unwittingly copies the viral DNA along with its own. The fact that the researchers found this in nearly 10% of healthy individuals perfectly mirrors the epidemiological data, which shows hepatitis B is highly endemic in Vietnam.
It is a literal biological footprint. The human genome is carrying the molecular ghosts of the viruses that have swept through that specific region over time. It shows that our DNA is a living historical document.
And it really is.
But I want to zoom out here, because this massive database, these 8.5 million novel variants, the global impact of this doesn't stop at Vietnam's borders, does it?
No, the geopolitical and global health implications are vast. Southeast Asia has a highly complex migratory history. Through admixture analysis, which basically traces genetic ancestry pathways, the researchers confirmed that these Vietnamese genomes share deep structural genetic ties with neighboring populations.
So mapping Vietnam with this level of resolution acts as a keystone for understanding the genetics of the wider region. Like, who do they share ties with?
We're talking about shared biological history with groups like the Chinese Day, populations in Singapore, Malaysia, and even indigenous groups in Japan.
Oh, wow.
And because of that, this new data dramatically improves the computational process called genetic imputation.
I've heard this described as predictive text for DNA. How does the algorithm actually guess the missing genetic data?
It relies on a concept called linkage disequilibrium. Essentially, genes that sit very close together on a chromosome tend to be inherited together as a block. So if a scientist only has an incomplete DNA sample from a patient, but they identify a few known markers, the algorithm looks at a reference database and says, okay, every time we see marker A and marker C, marker B is almost always between them. It imputes, or fills in, the blank.
But the algorithm is only as good as the reference text it has learned from.
Exactly. Before the VN1K Project, if an algorithm was trying to impute the missing DNA of someone of Southeast Asian descent, it was making guesses based heavily on European linkage patterns.
Which is like trying to autocomplete a sentence in Vietnamese using an English dictionary.
Right, the resulting errors were significant. But now, with this highly accurate regional pangenome data, the predictive text is vastly improved for millions of people of Asian descent all over the globe, whether they live in Hanoi, Paris, or Los Angeles.
By deeply mapping this one country, they have effectively sharpened the medical resolution for a massive portion of humanity. That is incredible.
It is a massive leap forward.
So let's synthesize this journey. We started with the medical mystery of a common seizure drug acting like a deadly poison. We watched scientists discard the flawed linear map of the human genome and build an AI-assisted pangenome graph. And through that, we uncovered 8.5 million biological secrets that had been hiding in plain sight.
And this research fundamentally dismantles the myth of standardized medicine. It provides three critical takeaways. First, utilizing a single European-centric genetic reference manual is dangerously inadequate for global medicine.
Right. It produces clinical blind spots that lead to misdiagnoses and toxic drug reactions.
Second, the VN1K Project proves that localized high-depth sequencing is an absolute necessity. It reveals the unique genetic traits, like those HLA variants and epigenetic methylation patterns, that dictate everything from drug reactions to liver health.
And finally, it proves that true precision medicine is mathematically and biologically impossible without capturing the full diversity of the human race. You just cannot heal a global population with a regional manual.
Exactly. Knowledge is only valuable when it's applied. These 8.5 million novel variants are a stark reminder of how vast and complex human diversity truly is.
Which leaves us with a rather profound final thought for you to mull over. If just one study in Vietnam can uncover 8.5 million biological secrets we didn't know existed, what other medical miracles or hidden dangers are quietly waiting to be discovered in the DNA of the rest of the unmapped world?
That is the million-dollar question.
Thank you so much for joining us in the Deep Dive Lab today. If you enjoyed uncovering these biological secrets with us, please make sure to follow and subscribe for more fascinating science stories. Until next time.